All right, here we are. It's Thursday, 4 o'clock on the West Coast. Adam, how are you?
Doing great. Excited to be here, Byron.
Yeah, we've got some people joining us. We're just going to wait a few minutes while we have a lot of people registered for this webinar today. So we'll just give them a few seconds, a few minutes here to log in, and got a lot of exciting information to share with them. And so delighted that you're joining us. And it's been quite a journey, quite a journey.
But we've made a lot of progress in
We
this journey
have. I
so
mean,
far.
last year was such a remarkable year of discovery, and now we're putting it all into practice. And we got so many people that are rooting for us and supporting us, and more coming on board. So excited about it. So we'll just wait for just a minute.
hmm
And I was thinking that with everything that's going on, there's never been a better time to find a better way to treat cancer.
Just the tools that are available right now and the pipelines and the technology, it's just a really exciting time to be in the cancer space.
Yeah, and one of the things we'll touch on briefly, and you can talk a little bit about if you like, is the role that artificial intelligence is playing in all this. I know that early on in your career, and I don't know if you were just prescient or if you had some sense that this was going to be a big deal, but you made the decision to become a computational cancer biologist, and boy, that was a good decision.
I consider that divine intervention, but it's been one of the best decisions I've made. Other than, marrying my wife, of course, but
Well, that's great. Well, listen, I think that yeah, here we go. So, listen, we want to welcome everybody for joining us today on the CancerVax webinar. We've been looking forward to this for a long, long time. We've got some fabulous and exciting information to share with you. We understand that we have a pretty diverse group of people that are joining us. In some cases, these are current investors, people who are looking to enhance their position in supporting CancerVax, people who have been following us for some time and are still trying to decide whether to make it part of their portfolio, and some people who are hearing about us for the first time. So we're going to try to satisfy all of those diverse audiences in our presentation today. Some of it will be repetitive, but we hope to make your investment in our time today very valuable. So welcome. Thank you for being here. We've got people from all over the country and quite frankly, internationally. And we really appreciate you taking the time to join us today. My name is Byron Elton. I am the Chief Executive Officer of CancerVax and a cancer survivor, I'm happy to say. I'm joined today by our principal scientist, Dr. Adam Grant, who I will give a proper introduction to in just a moment. Um, but let's start. You can see on your screen there, the reality is that cancer is the second leading cause of death in the world today. 10 million people die every year from cancer. And there's one main reason why that happens. And that is because the body's immune system, which is designed to protect us and does an extraordinary job in most cases, doesn't easily recognize cancer cells. It does an incredible job with things like measles and chickenpox, but not with cancer. Now, why is that? Well, the fact of the matter is that cancer cells aren't foreign invaders. They start as healthy cells, they mutate into cancer cells, and the body hasn't figured out a way to recognize those. And so our approach is very unique, and that is that we trick the body's immune system into thinking that cancer cells are pathogens that they already recognize, like measles, and tricks them into killing them. So I'd like to start off with showing you a quick 2.5 minute explainer video. This is on our website, by the way. And if you haven't spent any time there, cancervax.com, that's VAX.com. Great place to start. But here comes the video. Let's take a 2.5 minutes and watch this. I'm not hearing any sound. We might need to start this over again. We did this earlier and the sound was great.
CancerVax is creating a better way to fight cancer. The CancerVax way can simply be described as follows. 10 million people died last year because the body does not easily recognize and kill cancer cells. However, the body is very good at killing diseases that it recognizes, such as measles. Our unique technology disguises cancer cells to look like measles and tricks the body into killing them. That's it in a nutshell. That's the CancerVax way. How is that possible? Let me explain. Our technology, which we refer to as our universal cancer treatment platform, is packaged into injectable nanoparticles that are programmed to seek out only cancer cells. A single injection contains billions of CancerVax nanoparticles. Each nanoparticle is equipped with detectors to seek out specific cancer cells. The nanoparticles latch on to cells with the matching surface protein. The nanoparticle releases a smart mRNA payload into the cell. If the cell matches the genetic signature in the payload, then the cell is confirmed as the target cancer cell. The smart mRNA then tells the cancer cell to produce proteins associated with well-immunized diseases, such as measles. This makes cancer cells look like measles to the immune system. The body's immune system automatically attacks and kills cancer cells as if they were measles viruses. That's it. Our universal cancer treatment platform is a customizable platform that can treat many different types of cancer without harming healthy cells. We make cancer cells look like measles and trick the body into killing them. No one in the market is taking this novel approach. We've invented proprietary artificial intelligence and machine learning algorithms to find cellular signatures for detecting various cancer types. We're using tried and true mRNA and nanoparticle technology that made the COVID vaccine so inexpensive and effective. 2 patent applications have been filed to protect our novel intellectual property. CancerVax is creating a better way to fight cancer. The CancerVax way can be summarized as follows. 10 million people died last year because the body does not easily recognize and kill cancer cells. However, the body is very good at killing diseases that it recognizes, such as measles. Our unique technology disguises cancer cells to look like measles and tricks the body into killing them. We invite you to join the fight.
Okay, so again, that explainer video is up on our website. Would encourage you to spend some time there, learn more about CancerVax, the company. So let's talk a little bit about who we are and what we're doing. We are a preclinical biotech company. We're developing this universal cancer treatment platform. The platform, because it's not designed for one particular cancer, but for a variety of cancers, as we've mentioned. Very simply, it's a 3-step process. It is we detecting, marking, 2-step process in marking outer cell and inner cell, and then killing with precision only the cancer cells. That's important because, as you know, as traditional approaches like chemotherapy kills a lot of healthy cells along with it. So the ability to kill with precision is really, really super important. Immunotherapy is a hot area in this, and for years there's been billions of dollars spent on trying to teach the immune system into recognizing cancer cells. Very, very limited success. Again, and I know this is somewhat repetitive, but we intend to make the cancer cells look like measles, tricking the body into killing these disguised cancers. We really look forward to a day, and we envision a day when treating cancer will be an off-the-shelf, customizable injection to address all kinds of cancers. So they say that you're known by the company that you keep. If that is true, we are in really, really good shape. I want to introduce you to our scientific team, and including one who's joining us today. We'll start with Dr. George Kadabaugh. He's our Chief Scientific Officer. Dr. Kadabaugh has a very deep expertise in immunology and cancer biology and a track record of developing first in human therapies. He's full-time with us. Dr. Adam Grant, who joins us today, computational cancer biology, and by the way, the co-inventor of this platform. And he specializes in identifying the exact genetic singles signals that differentiate healthy cells from cancer cells, another very important part of our technology. You'll see Dr. Sumant Ramachanda there, seasoned pharma executive. He's led major R&D organizations in companies that you've heard of, like Pfizer and Baxter, and most importantly, has helped bring multiple therapies into clinical development. Dr. Gordon Ringgold, he's a serial biotech founder and innovator. His work has led to technology used in many, many therapeutic products. That brings us to Dr. Mark Davis. Now, I want to spend a little time with Dr. Davis. Feel free to Google him from Stanford. Dr. Davis is, and I don't exaggerate this, one of the most influential immunologists in the world. In fact, he discovered the T cell receptor. That's the foundation of how the immune system recognizes threats. At this point, I would tell you that in immunotherapy, lots of people hacking away at this problem. People like Dr. Davis get approached literally every day with companies wanting them to associate with them, to looking at their technology. Very few of them are successful. We could not be more delighted that Dr. Davis and people of his credentials have looked at what we're doing, seen the data, and said, I like this, I want to be part of it.
Yeah.
Lastly, we have on this slide Dr. George Kemble. He's a veteran biotech executive and a vaccine expert who's helped lead the development of FluMist. You may have heard of that, and many, many years of expertise and experience in translational medicine. So we have other members of our team. These are practicing physicians and scientists on our advisory board who bring many, many years of expertise. Most importantly, for those of you on this call, bringing drugs to the marketplace. So this is a team that we've assembled not only who study scientists, but they've actually brought these therapies to life. So along with a world-class team of scientists, we're working with specialized partners who support everything from our preclinical testing to mRNA development. These are the same organizations that support major biotech and pharmaceutical programs. People like Cytiva, Charles River, TriLink, Crown Bioscience, and Axis Bio. Again, world-class, best-of-breed clinical partners. Let's talk a little bit about the marketplace. It will not come as a surprise to you that this is a huge market. Cancer is one of the leading causes of death globally, millions of new cases every year. At the same time, the immunotherapy market is projected to grow dramatically. So 3 short years ago, less than $100 billion, $83 billion, projected in 6 years to be over $230 billion because existing treatments still leave major gaps in effectiveness and safety. And we really believe, we're confident that CancerVax will be one of those treatments. Okay, our platform overview. We're developing, as I mentioned, this treatment platform powered by artificial intelligence with all of that human experience behind it. Customizable, off-the-shelf injections to treat many, many different kinds of cancers. We talked about the detect, marking, and killing only cancer cells with our particular novel approach. And so if you want to looking at a label, we're in cancer immunotherapy or therapeutic cancer vaccines. It's the latest weapon in the war against cancer. It's a huge marketplace, and our approach is fundamentally different. This is really important to differentiate. We're not trying to teach the immune system how to recognize cancer. We're tricking it, and we believe that it's a better way to treat cancer. Okay, so I'm going to turn the time over to Dr. Adam Grant, our principal scientist, co-inventor of this technology, also a computational cancer biologist. Adam, thank you for spending some time with us today. I know your days are very, very busy and consumed with pursuing this technology, but could you just take some time and run us through some slides to further develop exactly what the platform is and how it works?
Yeah, love to, Byron. So glad to be here. So thankful for everyone that's taking the time to listen to us today. So, just want to go over a really high level overview of our platform and how it can be so customizable to different cancer types. So, the problem is, the body just cannot recognize cancer cells. And this is because cancer cells are derived from healthy cells. And as the cancer evolves, it's able to create signatures that hide itself from the immune system. And so it's just sitting there, and the immune system is all around it, but it just is not able to be activated. The security doesn't go off with cancer cells. And so, what and it's really sad, Byron. You mentioned that a lot of people are still receiving chemotherapy. That is the standard therapy for most cancer patients these days. And I mean, it's bad enough to have cancer, but I'm sure everyone has seen the effect that chemotherapy has, because what they're trying to do is they're trying to poison the cancer cells faster than the healthy cells. And but what we're trying to do is come up with a much more safer and efficacious way to treat cancer. And that's through our novel method of detect, mark, and kill. And so now we really want to talk about the lipid nanoparticle itself and how we are designing it to recognize cancer cells based off the surface proteins on the outside of the cancer cells, and then also look at the inside of the cell using our smart mRNA technology. So this way, we have a two-step process to specifically detect, mark, and kill only cancer cells while greatly sparing healthy cells. And so the first step is with the detection. And so we are designing a lipid nanoparticle that has a targeting moiety on the outside of it. And so, a lot of people are trying to do this, but what the community has realized is that there's not a single marker that is consistent across all cancer patients that is unique to the cancer cells. So no matter what, you are going to be, affecting the healthy cells as well. And so using AI approaches, we have identified some surface proteins on cancer cells that are highly biased towards cancer cells. And we're using that to make sure that our LNP gets into the cancer cells. But it's also going to get into the healthy cells. And this is by our targeting moiety on the outside of lipid nanoparticle will bind to the surface protein on that cell, and then the cell will grab it and release the mRNA inside of that cancer cell. But what's really cool about our platform is that this step two detection. And so we have utilized a lot of computational techniques, machine learning to identify optimal um, marker twos is what we call them. And this is part of our smart mRNA technology. And so when a cancer cell or healthy cell, uptakes our lipid nanoparticle, the mRNA is released inside of the cell. However, we are designing our smart mRNA to only be expressed in cancer cells, where it will be turned off in healthy cells. And we have some data that we're going to show with everyone that shows that. And so with this two-step process, we are able to much more fully select only cancer cells. And so that will allow our therapy to be not only more efficacious, but also to be much more safe to patients. So, they're not going to, I mean, the goal is to limit the off-target effects and the side effects as much as possible with our platform. But so it doesn't, so once the cancer cell has the mRNA on it and that it's going to be not turned off, it will express a part of the measles antigen or a pathogen, and that will come on the outside of the cell. And so, because of this, there's the immune system already can recognize, pathogens really well. I mean, everyone's been sick before, and the way that you get over that sickness is because your immune system says, "Hey, I got to get this out. I'm going to clear it." And so we're going to make the immune system do the same thing with the cancer cells. So once these cancer cells are expressing these antigens from a pathogen such as measles, it is going to recruit the memory CD8 T cells, and it will start attacking the cancer cells. And what's super cool about immunotherapy is that it doesn't stop there. It causes a huge cascade that brings in all of these other immune cells, and it elicits a the goal is to elicit a comprehensive immune response. And by all of these components working together, it's able to kill the cancer. And that's why in immunotherapy treatments, you're able to see such great responses. But now the problem is we don't know who they're going to benefit. But with the CancerVax platform, we're hoping that this will be much more universal because we're not dependent on certain biomarkers per se that they are. We're not trying to teach the immune system, like you said. We're going straight to the heart of the problem, which is the cancer cells, and making the immune system kill the cancer cells by tricking it. And so, just an overall, a brief summary is, we are going to make cancer cells look like a common immunized disease. We're using specific viruses that everyone should have a pre-existing immunity to, like measles. I mean, over 95% of the population has received a measles vaccination. And so they already have those memory CD8 T cells. We're using this two-step this method to detect cancer cells, which is going to make our therapy more efficacious and dramatically improve the safety profile, which is our goal. you know, by using this, lipid nanoparticle technique to detect, mark, and kill only cancer cells, and also machine learning techniques, is that we're able to design these really unique biomarkers that are unique to the cancer cells. And Byron, what makes me the most excited is that this isn't we're not breaking ground per se on this therapy, which is great as a scientist. The amount of time it takes to validate a technology such as like mRNA, it's just crazy. But it's already been validated in billions of people because of the COVID, shot. And so we're able to utilize the mRNA technology specifically for cancer. And then also, the lipid nanoparticle technology. We're working with Cytiva, who is an expert in the field and already has these lipid nanoparticles in the clinic. And so because we're able to, team up with them, is we're able to really expedite this process. So we don't have to validate these. These are already, in people. And, everything we do is we are patenting everything to, protect our intellectual property. You know, we talk to our lawyers all the time and are always showing the data that we get with them.
So speaking of patents, by the way, thank you for that. You did an extraordinary job of explaining that. Speaking of patents, intellectual property IP is so paramount for what we're doing, and we're working with some of the best IP attorneys, as Adam mentioned, in the country, to help us structure and protect the technology um, by creating what we call a patent thicket. And the reason it's a thicket is because it's
Mhm.
both offensive and defensive IP protection strategy. And we've already designed and validated our smart mRNA in cancer cells. We've built therapeutic nanoparticles. We've initiated animal studies to evaluate safety and effectiveness. So it's really important that we protect what we've done. Um, so go to the next slide there.
Yeah, and this is
Yeah,
really, oh, go
take this.
ahead.
This is exciting.
Yeah, sorry. Even when I just see the data, I get excited about it. Byron, sorry to say. But this is really where the rubber meets the road here. And so we just want to go over some of the results we received, like, within the last year, year and a half. And, a lot of these results are just, independent validations of our platform. But I mean, you'll see how they're all coming together to generate this universal treatment. So what we're looking at here is this is showing how our smart mRNA works in a cancer setting and in a healthy cell setting. So on the left here, you'll see these green cells. And so what's happening is that we delivered our smart mRNA into these cancer cells, and this was just a fluorescent mRNA called GFP. And the fact that they're glowing green means that our mRNA is being translated into protein and that it is in the cancer cells. So with our therapy, what would happen is that these cancer cells would be having the antigens on them, and then they would be able to be recognized by the immune cells. What's really cool is on the right side of this picture with the cells is that the green is not there anymore. And that's because we synthetically made these cancer cells have a signature that a healthy cell would have. And so it is turning off our mRNA, which means that we are able to use this smart mRNA technology to, control the expression of our mRNA payload in specific cell types. So we showed this synthetically, but on the right, you know, we administered the same mRNA into healthy like cells. And what you can see is on the left is that the GFP smart mRNA, the blue down there, the mRNA is not being expressed nearly as much as the control mRNA, which is, we call it the dumb mRNA because it's not able to recognize
Yeah.
the genetic signature of the healthy cells. And so what we're showing here is that our smart mRNA is doing exactly what we predicted, is that it's being expressed in the cancer cells, but being turned off in the healthy cells. Byron, I just want to say this was really exciting because this is one of the first experiments that we did here at CancerVax. And this was all these biomarkers or genetic signatures were chosen based off of AI techniques. And I mean, I'll be the first to admit it's a little nerve-wracking, the computer's telling me what to do, but by giving it all this data, looking into it, we were able to get it to work the first time, which was like so exciting as a computational biologist. And since then, this was just a test case. Since we've done this experiment, we have started to design smart mRNA specific for certain cancer types, like liver and pancreatic cancer, what you said. And we are getting close to validating, this smart mRNA technology in mice, right around the corner. And so those results are going to be very exciting to see.
So Adam, not only have you and the scientific team been pleasantly surprised and excited about this, but so have some of our developmental partners, haven't they?
Yeah, it's really great to see their reaction because we just kind of tell them what to do and they do the experiment and they come to us like what are you guys doing? How are you able to do this? We're just looking at things differently than anyone else is. And that's because of this mRNA technology that we're working with. Okay, so this next milestone, is involving our payload because, like you said, our goal is to create an mRNA that is able to make the cancer cells, look like they're virally infected and activate an immune response. And so, what this is showing here is that we were able to do that, in a petri dish per se. And so looking at this figure on the far left, we have our negative control, and this is just standard mRNA that is not virally associated at all. And so what we did is we made the cancer cells, we gave that mRNA, and then we added immune cells, and then we measured if those immune cells were able to be activated by our mRNA payload. So the mRNA as the negative control, which had nothing to do with viruses, was like, okay, the immune cells didn't react. And that's what we were hoping for. We we created two different mRNA payload, a measles payload and a SARS-CoV-2 payload. And the reason why we did this is because I mean, so many people have immune memory against measles due to the vaccine that they've got, and also to SARS-CoV-2 because of the COVID-19 pandemic. And this was just a random individual. We had no idea what their previous vaccination status was. We didn't know if they had COVID. We just chose this person. And what we saw is that this person's immune cells, they were able to recognize the cancer cells, and the immune system became activated. And they started getting angry and started attacking the cancer cells with this payload. And what was super cool is compared to the positive control, which is something that is commonly used in the scientific community. It's a combination of different viruses, is that our mRNA payload was working just as well as the positive control, suggesting that we were able to get a very strong immune reaction with this payload, or both of these payloads. And so this confirmed that our trick worked, that we were able to give this to the cancer cells, express the mRNA, and then it tricked the immune cells into attacking them.
Amazing.
Okay.
This is my favorite slide, by the way.
Yeah, it really just came out a couple weeks ago. It takes time, but we are finally put in the pipeline. We developed the LNP. So, yeah, sorry, I'm getting ahead of myself again. So what this is showing is that this is, our marker one technology. So if you remember, we have an LNP and we are putting something on the outside of the LNP to target only cancer cells, not healthy cells. And the marker is much more biased towards cancer cells than healthy cells. And we are comparing that to an LNP that doesn't have the targeting moiety on it, or there's nothing on the surface. And so what we saw is on the left here in the mice that's glowing. So this really big red dot right here is the liver. And the LNP that doesn't have our targeting moiety on it is practically 90% of it is getting into the liver. And this is really bad news because what happens is when it's stuck in the liver, it's not able to go through other organs and tissues and to be effective. And the liver drugs commonly get in the liver, and this is one of the major reasons why therapies fail. It's because of the liver damage that these drugs do. But amazingly, on the right of the mouse is when we added that, targeting moiety on our LNP, it was able to sneak by the liver. And this is what we were hoping for, but I'm going to be honest, we did not expect it to be this significant. And the fact that we're not getting stuck in the liver means that we're able to achieve this systemic circulation, that our LNP is able to go through the mouse's body and it's able to not have any liver problems. And the mice, they were completely fine with this therapy. They didn't show any signs of being unhealthy. It was just completely fine because they're not having this liver damage that occurs. And so this is just suggesting that our marker one technology is really, being more biased towards cancer. And so one of the next experiments we have is that we're going to be doing this where we're going to be putting a tumor in the mice and looking at the uptake. But this is the first thing we needed to see is that are we going to be able to escape the liver? And Byron, I'm going to tell you, we talked to a lot of people, a lot of partners for LNP development, and everyone just told us that, guys, you're just going to get stuck in the liver. Like, it's really problematic. But the way that we modified these LNPs, we were able to overcome one of the largest barriers of LNP. We still have to do testing, but this is the first step in a really promising direction, which was so exciting for our team.
I don't think we can overemphasize how big a deal this really is, getting through the liver.
Yeah, liver accumulation takes down so many biotech companies. It's so devastating. You spend so much time working on a therapy. You see that it works in the systems it works in a petri dish, great. It works in a mice, great when you're doing like intratumoral injection. But when you start doing this IV intravenous injection, you they see that you start having these problems. I've been a part of a biotech company where it the program got shut down because of an incident with liver failure from our therapy. So this is just huge for us of what we're seeing and the potential that we have to really be a universal platform.
Wow. Well, thank you for that, Adam. I continue to be impressed with your passion for this. I mean, I
Yeah.
know the kind of hours you guys put in on this and the experiences you've all had. Has there been anything even close to as exciting as this that you've ever worked on?
Oh, definitely not. I mean, this is for someone in a computational biologist. Working on this project, this platform is just a dream come true. And it's really because of the potential. And, Byron, so what we've done is we've really taken a strategic approach. And I hope, the people notice that we have started independently validating each of these steps. Our marker one technology, our marker two technology, our payload to trick the immune system. And what we're seeing is that we're getting great results in all of these. I mean, there are additional tests that need to be done, but we're getting very close to a point where we can start combining all of these into a single lipid nanoparticle and start evaluating it in a tumor setting, measuring the efficacy. So, I'm really hopeful that we're able to start generating a lot of data soon and start seeing how these different components are working together.
Yeah. Well, again, listen, Adam, thank you for taking time out of your day to be with us. Let me kind of bring this full circle. We've kind of come to the end of our time here. I live in Los Angeles. This is Dodger country. I was at a baseball game the other day and it was Stand Up for Cancer. And they sell it every game, right? 56,000, whatever it is. And at one point they say, if you've got somebody in your life that you want to say, stand up for cancer. Every single person in that stadium stood up. I dare say that there's nobody for whom cancer hasn't affected them in some significant way. And it's amazing to me that in a time where we have self-driving cars, I live here, Waymo, you jump in, they take you wherever. We're circling the moon as we speak.
Yeah.
And then when it comes to cancer treatment, we're still using a cancer drug that was developed when in the 1950s, chemotherapy.
Yeah.
Now they've gotten a lot better at it, let's be honest. But the fact of the matter is that many times, if the cancer doesn't kill you, the chemotherapy might, right? And so this idea that there has to be a better way to treat cancer is why CancerVax exists and why we're building this universal cancer treatment platform. We really believe that we have a better way to treat cancer, and that's what we're working towards. And so for everybody on this call that has already supported us, thank you, thank you, thank you. If you're thinking about it, we'd encourage you to make this part of your portfolio. We'd encourage you to go to startengine.com/cancervax. You'll see the offering and the way that you can invest in the company there. I would also say that if you want to talk to an executive at the company, call cancervax.com, a very simple calendaring calendar there that you can schedule a call with one of our executives. We talk to people from all over the world every day. This is extraordinarily exciting. We couldn't be more pleased with the progress that we've made. We're going to do another one of these on the 23rd, by the way, Adam. So, and we'll have more to share, obviously, as we go. So, we want to thank everybody for your time and your interest. We're excited about what we've accomplished. We're excited about what's ahead. And we look forward to continuing the conversation. Um, we did ask for some questions and answers. So we've got a couple. I'm just going to read them here. Well, we just had this on the slide. Somebody said, how much money have we raised so far? A little over 10 million
Mhm.
as the answer to that. Also, when does your start engine offering close? Oh, current round closes 21 days, May 1st. And then the last one for you, Adam. Are you ready for this one?
Okay,
I'll
hit
be
me.
anxious for you. This is a good one. I actually get this a lot. It's a good one. I don't think I've ever asked you this. This makes too much sense. How is it possible no one's thought about this before?
Yeah, it's fun because, sharing our story with people, it does. It just clicks. And the reason why this hasn't been done before is honestly, it's really because of the mRNA technology. It was just unfeasible. There, you just can't design a protein or, but with the combination of lipid nanoparticle and mRNA, we're able to really fully take this advantage of tricking the immune system. And I mean, COVID-19, it was such an unfortunate part of the world's history. So many people passed away, but because of that, we now have these tools that we have to better treat cancer. And so it's really, it's funny. I love Dr. Kataba. He always says that, a couple like 10 years ago, you said you were doing mRNA, people would think you're crazy, but now after being in billions of people, you're not crazy. You're just and and I will admit, at CancerVax, we have a really unique situation, not because of our scientific team, and I don't want to toot my horn too much, but with the computational techniques that we have. And, a lot of people don't realize this, but there's just so much data out there, but people don't know what to do with it. And so when we came up with this idea at CancerVax, we were able to mine all this data, use artificial intelligence to really optimize this platform and really turn it into something. And so, and sorry, I know I'm going on, but I just want to say one more thing to that question is that, and what's so cool is that a lot of these components of our platform, they have been already validated independently. And so we're really just taking the best parts of a lot of different, studies, technologies, and really merging it into this ultimate therapy or the potential of this ultimate therapy. So for those reasons, this is why we're at such a cutting edge, and why no one could have really done this before
Yeah.
until now.
Yeah, there's a reason for that. Yeah, we have, as we share this with particularly experts in the field or partners, we wait for that, what we call the aha moment, because they
Hmm.
hear all kinds of technologies and approaches and whatnot. And there's always one point in the conversation where there's kind of a moment of silence and they go, oh, okay. And there's the aha moment, right? So people
Well,
like,
and exactly. And we've been able to have moments like this with giants in the field, which is so exciting. I've been a part of those conversations, and these people have dedicated their lives to the scientific community. And when they are able to hear our story and what we're doing, they say, wow, that is really exciting, and I want to be a part of that. And we are, with our advisors, it's so amazing how much we're able to interact with them, how involved they are. And it's like, I cannot believe I'm able to be working with you know, Dr. Ramachandra, Dr. Ringgold, Dr. Davis, Dr. Kimball, and they are so invested in us, and it's very exciting.
well, thank you again for being with us, and thank you to everyone who joined us today. There has not never been, and there won't be a better time to invest and support in CancerVax. Again, startengine.com/, CancerVax. Seasoned investors will know that when this round ends, 21 days, May 1st, things change as new data comes in. And it goes without saying, this is a great time to jump in and support us, and we encourage you to do so. Thank you again for being here. Dr. Grant, thank you for being with us. And send you back to your computer in the lab. For your
Yeah, yeah.
so long, everybody.
Thank you.